right, people, let's start with the very top. So a huge chunk of this huge chunk of this topic is terminological. Okay, so like primary pathogen is the microorganism that causes the disease practically all the time, say salmonella. Opportunistic is the one that takes an opportunity. So things like Pseudomonas or Staphylococcus aureus, they're usually not really primary pathogens but can cause the disease in the immunocompromised patients. So resident microorganisms normally found, transient microorganisms just passing through. Now, body sites. So remember, there is a little list of body sites with microbiome and without. And there are a few question marks. So of those question marks, probably the most important one is the lungs. So generally we assume that lungs do not have resident microbiome. You can get microbes in there, but they are not resident. Well, obviously, body sites that are sterile are more prone to infection. So let's say if I ask which body site will be most likely to fall prey to bacterial infection, heart or skin, you can immediately say, of course, heart, because it's sterile, there's no competition. I hope it makes sense. Since I don't see your faces and all your microphones are muted, you can either sometimes unmute yourself and say, yeah, I got it, or just type something in the chat. So taxonomical composition at different body sites. At different body sites, taxonomical composition is different. Taxonomical composition at the same body site in different people is those taxonomical compositions are very similar. In health and disease, different of course, okay? So importance for the immune system development or obesity, I talk about it in the lecture, but the point is, we know that alterations to microbiome, say as antibiotic treatment, alters the development of the immune system and may lead to autoimmune disorders. The obesity, the microbiomes of mice or humans who are obese versus mice or humans who have normal weight, microbiomes in the gut are different. So can the microbial composition in one body site affect the physiology of another body site? The short answer is yes. We mentioned that when we talked about the role of microbiome in the autism spectrum disorder. So gut microbiome, specifically, immune system development. We mentioned that probably the normal, well, define normal, but gut microbiome heavily influences the development of the immune system in humans and in mice. Obesity, I already mentioned that's basically duplication here. So gut microbiome in obesity versus gut microbiome in organisms with normal weight, they are significantly different. It is association. We can't really say there is a causative relationship. Autism spectrum disorder, so it looks like certain probiotic species of bacteria, they can influence the severity of autism spectrum disorder. So that's that. Look, I'm not going to ask you to dig internet and give me a comprehensive review of role for gut microbiome in the autism spectrum disorder, but you have to be aware of things that I talked about in the lecture. So infection is when microorganism replicates. Asymptomatic, inapparent, well, that's that. No symptoms. Few things: communicable, non-communicable. So communicable, and we're talking infectious disease. So if I for some reason ask you to give me an example of communicable or non-communicable disease, I mean, heart disease is non-communicable, but it's not infectious, so that would be a bad example. So non-communicable diseases, um, examples can be peritonitis, or we talked about amoebic invasion, Naegleria fowleri, brain-eating amoeba. That's another good example of non-communicable disease. Now, nosocomial versus iatrogenic. Sometimes folks get confused. Nosocomial disease is the disease because you were in the hospital. That's it. You just were there. Iatrogenic is the disease that results from the medical treatment. So when patient acquires cytomegalovirus because he received or she received a donor kidney that was infected with cytomegalovirus, that is iatrogenic disease. So that is very specifically from the procedure. If you just go to the hospital and just stay in the hospital and acquire something that is not the result of the procedure but the result of being in the hospital, that's nosocomial. Zoonotic, coming from animals, or can come from animals. Same disease can be transmitted between humans and from animals to humans. An example would be coronavirus. Yeah, for some reason I have it twice. Never mind. Probably I just copy pasted it, so we got this all. Contagiousness, it's not really a formal term. It's ability of microorganism to infect. Now, basic reproduction number. What does it characterize? So, what you have to appreciate here. Basically, let's talk about the elephant in the room, the coronavirus. So basic reproduction number is estimated for coronavirus at about 3.5. Okay, what does that mean? It means that a single person over the course of infection will infect 3.5 people, okay, on average. It doesn't show you how severe the disease is. I have a table in the PowerPoint which shows that for Ebola, the basic reproduction number is 2. And for, I think, tetanus is like 7. Although, well, okay, tetanus is probably, for mumps, it's about 7. So mumps spreads through the community much faster. But it doesn't mean that it's worse in terms of the disease presentation. It's just how fast it spreads. Okay? I hope that is clear. So basic reproduction number only reflects the ability of microorganism to spread. And just FYI, just so you understand, it's the number that is modeled. And right now, if we will shut down if we will go into the complete lockdown like China did, the basic reproduction number will quickly fall under 1 because people simply won't be able to spread anything. Pathogenicity, virulence. So pathogenicity is qualitative, can or cannot cause the disease. Virulence shows you how bad the disease is. So all influenza viruses are pathogenic. They all can cause the disease. But say, according to our observations, okay, screw flu virus. Screw flu, let's talk about corona again. 3 coronaviruses, SARS, SARS-CoV-2, and MERS. All of them are pathogenic. But MERS is much more virulent. The disease that MERS causes is much more severe than the disease that any other virus causes. Properties on the site of infectious agent. So proper portal of entry is required. Infectious dose should be sufficient, and a variety of virulence factors. So virulence factor is any trait in the phenotype that increases the virulence. If we talk about bacteria, things like flagellum is the virulence factor. thing, antibiotic resistance. What else? I'm just trying to think about other antibiotic resistance, ability to produce toxins, presence of certain exoenzymes, ability to evade immunity. All those are virulence factors. Now, host-related factors. I have a whole slew of them lined up in the PowerPoint. So right here, you see on the right. But these are pretty obvious, you know, like things like age. So we now see that people who are older are more prone to the infection, or immunosuppression, or presence or absence of underlying conditions. Like now we see that people with underlying conditions like heart disease are more susceptible to die from SARS-CoV-2. So that's host-related factors. Have an idea what they are, okay? Median infectious, median lethal dose. You have 100 people, you give a median infectious dose, you can expect 50 people get sick. If you give them all median lethal dose, you expect 50 out of 100 to die, okay? Now, this, two questions, we discussed it with Demisha the other day. Can you compare the virulence of two unrelated pathogens like a virus and a bacteria using, you know, median infectious or median lethal dose? The short answer is no, you shouldn't. Let me show you. So here, take Vibrio cholerae here, right here, versus norovirus. Norovirus has much lower infectious dose, meaning that you need just a few particles to cause the disease. While Vibrio cholerae, you need 1 million bacterial cells to cause the disease. But if I would have to pick between norovirus and cholera, I would pick norovirus every day of the week, because the disease presentations are very, very different. However, going back to our study guide and looking at this.
My name is Can I get your first and last name, please? Demisha Varner. Oh,
Now, looking at this statement here, okay? So go back here. Look, you have enterotoxigenic or enteropathogenic E. coli, which have enormous median infectious dose, like millions or billions of bacteria. What they cause? They cause diarrhea. If we talk about enterohemorrhagic E. coli with infectious dose of 10 to 100, this microorganism is much more virulent. And it's diarrhea, dysentery, hemolytic uremic syndrome, all kinds of crazy stuff. So basically, we can say this, that the lower, regarding this statement, this one statement, the lower median infectious or median lethal dose, the higher the virulence. It's like the smaller amount of poison, okay? I hope it makes sense. Portals of entry, portals of exit. We don't talk about any, sorry, oh yeah, there was some kind of a glitch here. We don't talk about any particular specific portals of entry, portals of exit. So just know portals of entry is where microorganism gets in, and portal of exit is where it gets out, things like, you know, blood or respiratory tract or urine, stuff like that. What happens if microorganism use portal of entry that is different? Disease may not happen, or it may have different presentation. Acronym TORCH stands for, you don't need to know what it stands for actually. You need to know that acronym TORCH refers to the pathogens that can be transmitted via the the placenta. Adhesion happens first. Bacterial mechanisms for adhesion, they're listed. It's adhesins, fimbriae, capsule, slime layer, pili. Main viral mechanisms for the adhesion, viruses attach to the receptor. Infectious tropism, maybe some of you were puzzled with that word. Tropism means where infection happens. So neurotropic infections happen in the nervous system, hepatotropic in the liver. So receptor for the virus. If the receptor for a particular virus is found on the cells of the liver, this virus will infect liver, basically. That's the idea. Hepatitis C is an example. Invasion, getting into the tissue or into the cell, depends on the microorganism. Viruses use endosomes since they have to get into the cell and they large, so they often use endosome in the process of endocytosis to get into the cell. Virulence factors, defined already. So exoenzymes. 4 classes of exoenzymes: glycohydrolases, phospholipases, proteases, and nucleases. So those are 4 classes, how they can contribute. For instance, glycohydrolases can break down carbohydrates and destroy extracellular matrix. Proteases can break down collagen in the connective tissue. Nucleases can destroy DNA traps that are deployed by neutrophils to contain microbes. Phospholipases can break down phospholipid bilayer and lyse the membrane, leading to the cell death. So basically the idea is that exoenzymes somehow negatively modify the environment, and the environment for those microorganisms is essentially a cell or tissue. Cytokines store abnormal production of cytokines, abnormal inflammation that leads to systemic inflammatory response and shock, toxigenicity, ability to produce toxins, toxinosis, disease caused by toxin, toxemia, toxin in the blood, intoxication is the ingestion of the toxin. Exotoxins. So let's go through this step by step. Endotoxins, only gram negatives. Those endotoxins are the molecules, LPS, lipopolysaccharide molecules, that are found in the outer membrane of gram-negative microorganisms. Exotoxins can be produced by both gram-negatives and gram-positives. In terms of the composition, endotoxins, lipopolysaccharide. Exotoxins, usually proteins. Exotoxins have very, very specific mechanisms of action. You may have noticed there's a huge table with the list of exotoxins, such as botulinum toxin, that toxin that's produced by Clostridium botulinum, which inhibits the release of acetylcholine, neuromuscular junctions. That's very, very specific mechanism. 3 main classes of exotoxins: intracellular targeting toxin, membrane targeting toxin, and superantigen. So the question that I guess some of you may have: do we need to memorize that entire table? The answer is, of course not. Unfortunately, we were not in class, otherwise we would discuss some of those toxins because they are absolutely fascinating. But what you really need to know, you need to know the categories, okay? You see? So you should be able to tell me, what is general mechanism of superantigens, okay? Overstimulation of immune response, basically. So that mechanisms, they refer to this category story. Effects of endotoxins. Endotoxins promote non-specific inflammation in the host. So we have myalgia, fever. Sometimes they are referred as pyrogens. Just FYI. Pyrogen. molecule that can generate temperature. So endotoxins are extremely heat stable, and exotoxins are not. Endotoxins have very high median lethal dose. Exotoxins have low. So exotoxins, you need smaller dose to see the pathology. And exotoxins can stimulate the production of production of antibodies, basically. So if you take an exotoxin and stick it in the patient will produce antibodies against it. Now here, why it is important to detect the endotoxins in the sterilized fluids. Imagine you have water for injections, and it is absolutely sterile. There are no endospores or bacteria, but as the result of death of gram-negative microorganisms, you have endotoxins, still have endotoxins in it. If you inject this fluid, water, whatever, into a patient, endotoxins will go into a patient, and patient will develop really severe high fever, myalgia, inflammation, blah, 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 blah. If patient is already sick, so the patient may die of that inflammatory response. So you use LAL test to detect endotoxins. Basically the idea right here, and I'm going to take a pause after that. The idea of LAL test that is used now is that endotoxin activates a recombinant enzyme called protease. Here. Okay, so it activates protease. The more endotoxin you have in a sample, the more protease is going to be activated. And this protease, the enzyme, breaks down so-called chromogenic substrate. So when this molecule is broken down by the protease, it starts to produce luminescence. It starts to shine, quite literally, shine. And the more toxin, the more protease, the more light, you can build a standard curve, you can find your the concentration of endotoxin in your sample. So it's really quantitative and convenient method. Any comments, anything that is not understood? I'm gonna Darla, I'm gonna unmute you for a second. Are we doing all right?
Oh yeah.
Oh yeah.
Very good. Amy?
Doing fine.
Doing fine.
Okay. Nicole, welcome. Are we doing all right?
Yep, we're good.
Yep, we're good.
Awesome. So, I mean, if I didn't unmute you, you can unmute it yourself, like Olivia or Amanda. If you want to say something, don't hesitate. Or you can just write into the chat. Okay. Okay. Now, avoidance of immune response. So capsule, bacteria and fungi protect microorganisms from phagocytosis. Proteases. Bacterial proteases, exoenzymes can be released and can destroy antibodies. So we got capsule, got proteases. Intracellular infection, especially infection of immune cells, like Mycobacterium tuberculosis that infects macrophages, basically hides itself under the nose of the immune system. Inside of the cell, it's much harder to mount an immune response to intracellular infection. Molecular mimicry. Microorganisms somehow make themselves look like the host's proteins or whatever. So I give an example of, I believe, schistosoma doing that. Complement inhibition. Complement is one of the innate immune responses mechanisms. So if you inhibit complement, it protects you. Leukocidin specifically, mostly Staphylococcus and Streptococcus, they basically kill white blood cells. viruses very often inhibit the innate immune response. And the Red Queen paradox is the fact that, pathogens in humans co-evolve, and pathogens, the evolution of the pathogens is to avoid human immune response, avoid being shut down. An evolution of a human is to actually shut down the pathogen. Okay? So when microorganism and a human, they co-evolve, they put, basically the evolution drives them to develop new mechanisms just to stay in the same ecological balance. Because you can imagine that for a microorganism, it's not really beneficial to kill all the hosts. Signs and symptoms. Signs are objective, symptoms are subjective. So like fever, actual fever, like 104 degrees or high blood pressure is a sign. Something that can be like rash is a sign, right? Nausea or headache is a symptom. 5 main stages. Incubation. You don't see any signs or symptoms of the disease during that stage, but microorganism starts to replicate. Prodromal stage, when you feel kind of the actual like disease stage, when you're really sick. Then unfortunately sometimes death happens, or the person may go in the decline stage and convalescent stage at the end. So convalescent stage is when you are basically recovering, like you're not really sick, okay? And actually coronavirus provides a really good example what happens. So it turns out it's almost certain now that there is a transmission during incubation period, okay? So during incubation period, microorganism starts to replicate. Then prodromal stage and disease stage for coronavirus, it replicates more and more and more. Then if you are lucky, when you start to recover, the amount of virus in your organism during the decline stage and convalescent stage goes down. But it was shown that even during convalescent stage, people can still shed virus. So this graph that you can see here, it's pretty applicable to the coronavirus, but you have to appreciate that for some infections, it's not true, okay? Meaning that you can have all the replication happening in the very beginning, and then when disease happens, the virus is practically gone, or bacteria is practically gone. Localized focal systemic. Localized at the site of infection, sore throat. Focal migrates from the original site of infection to one another location. So you get from strep throat to endocarditis. Systemic, multiple organs are involved. Emia means in the blood. Bacteremia, fungemia, viremia, septicemia. Not just bacteria in the blood. Usually it refers to bacteria. Not just bacteria in the blood, but bacteria actively replicating in the blood. Primary, secondary time. You have first infection, and then due to the first infection and decreased immunity of second infection. Now, these 4 terms here, latent, acute, chronic, and persistent. So let's actually start with an acute. Acute means short-term, very well manifested infection. Influenza is an example of an acute infection. It's always acute infection. It hits you hard, you get sick, you recover, virus is gone. Chronic infection. Microorganism lingers on for a long time. That can be leprosy is an example of chronic infection. Hepatitis B or hepatitis C is an example of chronic infection. Usually in chronic infection, the symptoms are a bit muted, maybe, a bit blunted. They're not as pronounced. Latent infection. microorganism does not replicate and does not cause symptoms. Herpes. It has latent stage, and sometimes it reactivates. When it reactivates, it's an acute infection, but between the reactivations, it's in the latent stage. Persistent is an interesting phenomenon. So persistent infection is the one when microorganism does replicate. You can find it in, say, urine, but it causes no apparent symptoms. So there's a virus called BK virus, which does exactly that. So, for instance, you can find BK virus in the urine of pregnant women, but there's no pathology for pregnant women whatsoever associated with that virus. Sequelae, consequence of the disease, like neurological consequence. Reservoir versus host. So I want to delineate like this questions very clearly. So reservoir is something that is absolutely necessary to maintain the infection. Host is the organism that is commonly infected and gets the disease. The boundary is a bit diffuse, I would say. But for example, cholera, Vibrio cholera. The reservoir is brackish water. Bacteria happily grows in the brackish water. The host is the human. You drink the brackish water, you develop the disease. Another example, measles. It is exclusively human disease. Not any other animal, as far as we know, normally gets measles. So for measles, humans are both reservoir and the host. We good so far? I guess so. I'm gonna ask this question again. I hope that you can figure it out. I think one of the practice questions addresses this host versus reservoir versus possibly vector. That's like a big wink here, like know that stuff, be able to analyze what's what in a certain description of the disease. Vector, usually it's an animal, usually an arthropod, like a mosquito or tick, that transmits the disease. Mechanical vector transmits the disease, well, mechanically, from one, point to another, just carrying on its body, or just mechanically carrying the disease. And in biological vector, the microorganism actually replicates inside. So like dengue virus, mosquito is a biological vector because dengue replicates. Carrier, a human that can carry the disease. There are 5 major types: incubation, chronic, asymptomatic, passive, and convalescent. So some may ask, what's the difference between incubation and asymptomatic? Incubation carrier will be sick. They're just not sick now. Asymptomatic carrier will not be sick. They just don't know. Okay? Fomites, inanimate objects that can carry microorganism. Vehicle. Vehicle is not an object, it's a material. So, let me explain. This is a fomite. And coffee is a vehicle. Okay? Direct transmission routes. So we've got contact, and that's actually a great moment to mention this, because we're talking about transmission routes here. So one is contact, touching, via, like, if you get in contact with somebody's bodily fluids, when you kiss somebody, having sex falls into that category as well. So that's contact route. Now, respiratory route. This is really complicated because, for instance, right now, as I talk or I breathe, I produce aerosols. They're very fine with a very, very small size droplets. So that is truly respiratory. So basically, for SARS-CoV-2, the idea that like the not the dogma, but what is supported by the data is that people rarely transmit this disease by breathing. Okay? The main transmission is when people sneeze or cough. And when you sneeze or cough, there's actually a very a huge range of the size of those little droplets that are generated. I mean, when people sneeze, they expel a lot of mucus and stuff, and it can be pretty large chunks. So often this sneezing, coughing, I mean, it's not truly a respiratory route. It's a different route. Many people put it into the contact rather than respiratory. So basically, when we say that Ebola is not transmitted by a respiratory route, it means if we are sitting next to each other on a plane and I have Ebola virus, you're going to be fine. Okay? If I have measles, then you're not going to be fine. So we've got respiratory, we've got contact, vertical, from mother to child, either through placenta or during the birth, and vector-borne. I want to highlight when we say direct transmission route, we refer to the transmission from person to person. So yellow fever is directly transmitted by biological vector between humans, from human to human. West Nile virus is not transmitted from person to person at all. Just no direct transmission. Indirect transmission. Fomites, obviously. Vehicles. And respiratory droplets. So that refers to a fairly unique class of diseases. Let's say rabies. Usually it is transmitted from animals to humans when animals bite humans. But if you walk into a cave where there are bats carrying rabies, you can simply acquire this disease because you inhale the aerosolized feces of bats, and that inhalation often is enough. Dose is large enough to give you rabies. So it's like it's indirect route. You can't, like if I have rabies, again, on the plane, nobody's going to get infected. I can't sneeze on someone and give the person rabies. I'm going to pause for a second, and I'm going to okay. Mandy, you good?
Yes, I don't know how that happened.
What, I unmuted
Did you do that?
I unmuted you.
Yeah, I'm good.
Okay, I'm just asking that.
Yeah, my kid just keeps saying he wants to talk to my professor.
Yeah, well, I'm not really good at
You know?
talking to little kids, and I'm even worse with big kids. So, I mean, Olivia, we're good. Okay. Don't hesitate to unmute yourself and stop me if you need to ask a question. Okay, so we got this 4 steps of linking the infection disease using Koch's postulates. So you've got to isolate microorganism from the sick but not healthy. You have to be able to culture it in a pure culture. Organism has to produce the same disease in the model organism, and you have to be able to isolate that same microorganism from your model. It's really hard because sometimes you can get asymptomatic infections, so you're going to have microorganism being present in people without any symptoms. Sometimes you can have microorganism you can't culture it. Sometimes presentation of the disease in the model, like in mice, is different from people, so there are a lot of obstacles. This is why it's hard to apply. Molecular Koch's postulates basically link, they more refined, say they link the virulence to a particular virulence factor. So you find a gene that is different between the virulent and non-virulent microorganism. Okay? And then you knock out this gene, basically get rid of this gene, and if virulence declines, yeah, that's your virulence factor that determines basically the virulence. Like, there's an example in the lecture of enterohemorrhagic E. coli. You knock out the gene for Shiga toxin, it's not enterohemorrhagic anymore. You add this gene, enterohemorrhagic again. Okay. Incidence, prevalence, morbidity, mortality. Okay, that's really important. Incidence. Number of cases over a certain period of time in a certain population. Okay? You probably can see my screen. So this is the incidence, okay, of coronavirus in the United States. So if we assume that we've got 330 million people, then the incidence of coronavirus is a little bit more than 1 person per 1,000 of population, right? So 0.1%, okay, are affected. We're good, so I hope we're good so far. Prevalence. The percent of population that was actually affected by that disease. Not at a given moment, but overall. So, well, actually, it's not really right, because that's total confirmed. So that would be prevalence. The incidence right now would be that number minus 22,000, because they're not sick anymore. Okay? Yeah, here we go. So, for instance, you can say, what's the incidence of HIV in the United States last year? 2,000 cases. What's the prevalence of HIV? I don't know how many million people have the disease, right? Morbidity, percent of people in which disease actually causes, in which infectious agent causes the disease. So, for instance, if we take common cold, many people are infected asymptomatically, the disease is very mild, the morbidity is low. If we take Ebola virus, that's the opposite end of the pole. Morbidity is enormous. Virtually every person becomes sick, and many of them die. Now, this is the most interesting question. The mortality versus case fatality ratio. Let's go back to that fantastic immunological illustration right here. Let's talk about the United States, because quite frankly, United States don't care about anybody else in the world. So we have, well, about 13,000 deaths so far, right? And we have 400,000 confirmed cases. So, come on, dude. So that's calculator, my calculator. So I'm going to do so the current case fatality ratio is 3.2%. Case fatality. So that's the number of deaths divided by the number of confirmed cases. Okay? Now, does it actually reflect the true mortality of the disease? The answer is no, it doesn't. Because there are people walking around who had mild infection, didn't go to work for a couple of days, and they were fine. So the good news is the true mortality, or sometimes it is referred to as infection fatality ratio, is much lower. So there are estimates that for coronavirus, again, there's a great saying, every model is wrong, but some of them are useful. So folks from Oxford, I think, they modeled that the actual IFR for coronavirus is about 0.1%. So 1 out of 1,000 people actually die. 1 out of 1,000 actually infected people die. So it's kind of good news. Okay, sporadic infection that occurs from time to time. Endemic, like plague. Although plague is kind of tricky. Okay, let's say botulism. Okay, that's truly sporadic. Endemic regularly occurs in a certain region. All right. So Rocky Mountain spotted fever. Regularly, you saw this question on the quiz. Actually, I'm just lazy. I'm picking the question from the quiz. RMSF happens regularly in Missouri, Arkansas, this state. Is it endemic there? Absolutely, yeah. It's like every year, a few hundred people gonna get it. It's not like every year we have a few hundred people. Fine. If out of a sudden we have, instead of 100 people, we have 5,000 cases, then it's an epidemic. Okay. So I have this graph showing here the visits to emergency department due to influenza. Okay, you see in 2007, 2008, it was much more than usual. That was an epidemic. Okay. Finally, pandemic, worldwide distribution. I mean, if you think about it, we have several pandemics going on right now. HIV, tuberculosis. Virtually every country is affected by these diseases. Okay. Amy, we're doing fine?
Yes, actually my cat.
Yes, actually my cat
Okay.
unmuted me.
No, it's I unmuted you.
Oh, okay, I'm fine.
Oh, okay, I'm fine.
Nicole, we good?
Yep, we're good.
Okay.
Yep, we're good.
Darla, we doing all right?
We are great.
We are great.
Okay, chemical barriers. Low acidity, low pH, immunoglobulins, dermicidins, short chain fatty acids. Know where they are. Like, don't try to convince me that you can find dermicidins, which are normally present in sweat, like in the vagina. Last time I checked, it doesn't work this way. Mechanical barriers, well, dry skin and mucus. Mucus is one of the most important, okay? So, vaginal pH. What can change the pH of vagina? Actually, antibiotic treatment may kill lactobacilli, which leads to elevated pH, which makes vaginal mucosa more prone to the infections. Cilia in the respiratory tract. Cilia are destroyed, mucus is not moved, which makes the getting rid of the pathogens complicated. Innate immune response, very fast, local, not very effective, does not require previous exposure, non-specific. Now, look, if you really don't mind, this one, okay, I really don't want to recite the table. We've got it here, okay? I mean, yeah, let me explain. You don't have to worry about the structure. That I can guarantee. I'm not going to ask you like multi-lobe nucleus, acidic state, screw that. What I do want you to know, the targets and the basic mechanisms. Now, if I ask you, if for some reason you get a question and you have to describe basic mechanism of neutrophils, and you forget to mention respiratory burst or NETs and just say phagocytosis, I'm fine with that. If you completely in the wrong, like you tell me, okay, neutrophils can produce antibodies. No, they can't. Okay? Or you try to convince me that NK cells and lymphocytes, NK cells and lymphocytes in general, can phagocytose? No, they can't. So like the big chunks, like neutrophils and macrophages, phagocytic cells, eosinophils, cytotoxic cells, basophils, pro-inflammatory cytokines, lymphocytes, innate immunity cause apoptosis, stuff like that. Okay, I hope we're clear. Okay, so basically got this. Okay, just know that natural killer cells here are lymphocytes, but still they're not very specific, so they belong to innate immune responses. So, response to the injury. There is a tissue injury. In response to injury, cells release inflammatory cytokines. Inflammatory cytokines have several effects. They cause leukocytosis. Okay, they increase vascular permeability, and they stimulate production of adhesion molecules on the blood vessels, on the endothelial cells. So leukocytosis means a lot of new white blood cells. Increased vascular permeability allows plasma and white blood cells to migrate from the blood vessel into the tissue, and the adhesion molecules on the surface of the blood vessels, they basically trap white blood cells at the site of injury. So when white blood cells interact with adhesion molecules, they adhere to the endothelium, and then they undergo diapedesis, which means they migrate into the tissue. Two main types of the cells that play a role in such inflammatory response are macrophages and neutrophils. And basically adhesion molecules is how they find the site of the tissue injury. Once they are in the tissue, they migrate towards the exact injury site, the infection site, following the trail of inflammatory cytokines and chemokines. We call it positive chemotaxis. Monocytes differentiate into macrophages when they stimulated, when they are exposed to inflammatory cytokines or chemokines. So inflammation triggers the differentiation. Plasma leakage washes away the dead cells and cellular debris. Clotting factors trap the microorganisms in place and don't let them spread. Pathogen-associated molecular patterns are the molecules found on the surface or inside the microorganisms, the pathogens. So we talked about LPS, the endotoxin, before. That's the pathogen-associated molecular pattern. Why it's molecular pattern? It's found in all gram-negative microorganisms. So it is a chemical signature of gram-negative microorganisms. And these pathogen-associated molecular patterns that are found on the pathogens are recognized by pattern recognition receptors, which are found on the host cells, either on the cell membrane or endoplasmic membrane, endosomal membrane. And I mentioned in the lecture that which ones are on the endosomal membrane. So pattern recognition receptors on the endosomal membrane recognize viruses because endosome is intracellular and viruses are intracellular. So toll-like receptors is the class of pattern recognition receptors. So they belong to pattern recognition receptors. They can be found in a cytoplasm or endosomal, in a cytoplasmic membrane or endosomal membrane of the host cell. I answered that question because viruses are getting into the cell through the endosome, and usual effects is activation of innate responses inside the cell. Phagocytosis, relatively non-specific. So phagocyte, using its own PRR, recognizes PAMPs on the pathogen surface. So pathogen pumps bind to PRRs on the phagocyte surface. Phagocyte in response forms pseudopodia. Pseudopodia engulf pathogen and form phagosome. Phagosome fuses with lysosome, forming phagolysosome, and acid and reactive oxygen species and hydrolytic enzymes kill the pathogen inside of the phagolysosome. Two types of phagocytic cells: neutrophils, macrophages. Neutrophils die, they kamikazes, and macrophages don't. Okay, they survive phagocytosis. Complement set of proteins in the blood can be found in the blood, plasma. 3 complement pathways. So mechanism of activation of classical pathway of complement. Antibodies on the surface of the pathogen. Lectin pathway. Mannose sugar on the surface of the pathogen interacts with lectin, activates the lectin pathway. Alternative pathway. Various macromolecules on the surface of the pathogen. So what do you need to know? Do you need to know the intermediate steps? No. You need to know 3 mechanisms that activate 3 different pathways. You need to know that all those pathways converge on the same molecule, which is C3b convertase. Okay? So we've got lectin pathway, classical, alternative. So they converge, and you need to know the outcomes. The outcomes would be increased inflammatory response, increased phagocyte activity, membrane attack complex. We talked about membrane attack complex in the lecture. Formation of a pore, illustrated right here. And now opsonization. So opsonization, I had some questions about it. So what is opsonization? Proteins of complement C3a, for instance. Proteins of the complement can cause opsonization. They can stick to the surface of the pathogen, stimulating the phagocytosis. I hope that makes sense. Okay. So think of it this way. If you buy some pretty bland food, you may want to add ketchup or soy sauce to make it taste better. So opsonization is essentially adding ketchup or soy sauce to the pathogen so macrophages or neutrophils can recognize it better and destroy it faster. So I hope that helps. Interferons, chemically proteins, major types, 1 and 2. Interferon production is triggered by infection. So basically you have a cell that's infected, it produces interferons. Interferons are released from the cell. They can act on the same cell, that's autocrine effect. They can act on adjacent cell, that's paracrine effect. And they can enter the bloodstream and act on the distant cells, that's endocrine effect. And on the cell, they interact with the receptor. That's important because they are proteins. They can't cross the membrane, so they have to interact with the cellular receptor. And the effect that interferons produce on the target cell is that they basically warn it that, infection is somewhere in the body. So they stimulate various antiviral, antibacterial responses in the cells. In this picture here, so you see, this cell is infected. These cells, well, except these cells are not. So interferon mounts antiviral responses here, may lead to apoptosis, or may activate some immune cells. So it's basically a warning molecule rather than really like a treatment. Not a treatment, sorry. Doesn't like really lyse the cell or something. Interference can be used for the treatment, limitations, side effects. Okay. Another pause for 2 reasons. Amanda, you fine?
Oh,
Yeah, I'm good.
Okay.
yeah, I'm good.
Darla?
Yep, I'm good.
Yep, I'm
Anybody have
good.
any questions? Okay, I don't think so. Now, can you give me like 2 minutes? I'm going to run upstairs and grab a glass of water. Okay? Thanks. I'm going to pause the recording now. All right, I guess, yeah, recording is going on again. All right, adaptive immunity. So adaptive immunity, what are the unique features? Good things: specific, systemic, highly effective, forms immunological memory. Kind of not so good features. Takes a lot of time to develop and requires an exposure to a pathogen to actually mount. So cells have to interact with the pathogen. So that's kind of explains the difference, Innate is faster, adaptive is more specific. Innate is local, adaptive more systemic, so on and so forth. Okay, humoral cellular. Humoral responses mediated by antibodies, cellular mediated by T cells. So humoral response essentially B cell response, cellular response is T cell response. B and T cells anatomically originates in the bone marrow. B cells, B lymphocytes, mature in the bone marrow. T lymphocytes mature in thymus. And after maturation, they all populate lymphoid tissue, lymph nodes, spleen. And when they are exposed to the antigen in the lymphoid tissue, they differentiate into effector cells. Those effector cells being for B lymphocytes, plasma cells; for T lymphocytes, it's helper cells or cytotoxic lymphocytes or regulatory cells. And the effector cells actually elicit the response. So for cellular branch of adaptive response, helper cells activate B cells or macrophages, and killer cells cause apoptosis of virus-infected or cancerous cells. For humoral branch, you have antibodies. Plasma cells produce antibodies, and antibodies do a lot of stuff that we're going to talk about. Antigen. Complete antigen is the large and complex molecule that is foreign to a host. So, for instance, for me, rabbit's proteins are going to be antigens. For rabbit, my proteins are going to be antigens. Molecules that can be an antigen, protein, but be complex carbohydrate, or DNA. If a molecule is large but not complex, like polypropylene or something like that, it's too small, it's too simple, it cannot be an antigen. Antigenic determinant is the part of an antigen that is recognized by the immune system. So it's really important to understand. A part of an antigen that is recognized by the immune system. Like when you describe someone, when you describe a person, you don't say, this person, 2 legs, 2 arms, a head. You say, okay, really long nose, or, I don't know, very tall, a tattoo on the left arm, stuff like that. So yeah, we figured this. I don't think I talk about haptene, so we can screw that. I think it's like a remnant of the old story. Yeah, I don't talk about haptene, that's fine. So antibody chemically, so forget about haptene, and it can be gone. Antibody chemically, it's a protein, 4 building blocks, 2 light, 2 heavy chains. So function of the FAB region. FAB region, antigen binding, AB. Okay? So FAB region of an antibody binds an antigen. More specifically, FAB region binds a specific antigenic determinant. That's, of course, a variable part because it has to be changed. FC region binds various cells of the immune system, so C stands for either cellular or constant, whatever you prefer. This part of the molecule is more constant. B cell receptor. Okay, B cell receptor is the antibody. So let's talk about the populations and stuff. So you have a bunch of B cells that carry the same B cell receptor, structurally identical. That's the B cell clone. All those individual B cells together are called B cell clone. When they develop, the negative selection removes all the B cells that have B cell receptor able to recognize self-antigens. So we basically remove reactive B cells. B cells that can cause autoimmune condition. So when B cells are exposed to the antigen, only the population that carries B cell receptor. Okay, let me draw it. I will try to draw it really quick. So that's B cell, okay, that's B cell receptor. That's another B cell with a different receptor. So if you have an antigen like this antigen will bind to the B cell receptor on that bottom B cell because it fits here. Okay, I hope it makes sense. So this clone will be selected. That's a clonal selection. As the result of that clonal selection, B cells proliferate, so they become plenty, and they differentiate into memory B cells and plasma cells produce antibodies. So sometimes B cell activation can happen by an antigen itself. It's called T cell independent activation. And sometimes T cells are required, then it's called T cell dependent activation. VDJ recombination, it's somewhat hard to, well, you have a huge number of antigens, so to have a significant variety of B cell and T cell receptors, what happens is you have basically 4 construction parts, V, D, J, and C parts, okay? So, like on this image right here, so you pick one V segment, one D segment, one J segment, and one segment for constant region, and then you combine them. Having multiple variable segments, multiple diversity, multiple joining, and multiple constant regions, you can combine them like you combine, I don't know, like, I mean, it's a lot of combinations, okay? Number 27, number 13, number 2, number 1. That's one combination. Number 4, number 26, number 1, number 2. Different combinations. So all in all, that gives you trillions of combinations. Basically, your B cells can recognize any antigen whatsoever. 5 major antibody types. Right, let me clarify. You don't need to know percentage. You need to know if it crosses placenta. You need to know if it activates complement. Fixes complement means activates complement. You don't need to know what FC receptor binds to. FC fragment binds to. You need to know the general function, like immunoglobulin D, B cell receptor, immunoglobulin E, allergic responses, immunoglobulin A, dimer that can be found in the mucosal membranes and in the mother's milk. Immunoglobulin M, first response antibody is produced first and can fix complement. Immunoglobulin G can activate complement, can cross placenta, the main antibody of a secondary response. 2 main cell types, memory and plasma. Temporal antibody profile of the primary response. So here I refer to this image. So you see primary response, you have IgM first, IgG second. And then when you're exposed to the same pathogen, IgM and IgG coincide basically immediately, and IgG rises to much higher levels. So in secondary response, IgG is the primary type. Okay, we got this. Yeah, secondary humoral response is much faster and more robust. 4 types of humoral immunity, should be able to identify it. Adaptive, not adaptive, sorry. Artificial versus natural, passive versus active. Artificial versus natural usually doesn't pose any problem. Passive versus active. In active, you actually have an antigen that your immune system is exposed to. So like vaccine or infection, it's an example of active immunity. Immunoglobulins going across placenta into a baby, that's an example of a passive immunity. Major mechanisms, they listed here. Neutralization, usually viruses, blocks their binding to the cellular receptors. That's, by the way, if you've heard, FDA approved the use of convalescent serum for treating COVID-19. So basically, people who recovered, they give blood, labs isolate antibodies from the blood of those people and use these antibodies to treat those who are ill. So basically, it's a passive artificial immunity. Agglutination, binding multiple cells together, clumping them, so bacteria often agglutinated. Precipitation, binding soluble antigens, so toxins can be precipitated. Antibodies linking adaptive and immune responses. So a few things here. Let me go back here. So one link, activation of complement. So antibodies can activate complement. Okay, that's first link. Second link, you see that opsonization here? It's another link. So opsonization can be carried out not only by complement proteins, but also by antibodies. So when antibodies bind to the surface of the pathogen and attract macrophages, they play the same role as complement proteins do. So both complement and and antibodies can opsonize a pathogen. Okay. Nicole, we're doing fine?
Yep.
Okay. Mandy, we're doing all right?
Good.
Okay,
Yeah.
awesome. Amy, any questions?
Doing fine.
Doing fine.
Awesome. So, wow, that's like a podcast. Okay, major histocompatibility complex. So for our intents and purposes, things to know. T cells, here, T cells are not activated by the antigen itself. They have to have antigen presented to them. Antigen is presented by antigen-presenting cells. Antigen-presenting cells carry on their surface both type 1 and type 2 MHC molecules. So the role of MHC, major histocompatibility complex, is to present antigen. So those antigen-presenting cells carry type 1 and type 2. Okay? Now, in addition to antigen-presenting cells, you can find type 1 on every other nucleated cell, except for red blood cell, which is not nucleated, or platelet, which is not nucleated, or sperm cell. So, the role of type 1 in non-immune cells, role of type 1 MHC is to wave the flag saying, I am fine. Okay? I'm just curious if I can make myself a little bigger. All right, so like this cell is waving here. There's a little me in the corner of the screen. This cell is waving here. The blue flag saying, yeah, I'm okay. That's a liver cell, I'm okay. If liver cell becomes infected with the virus, it starts to wave the part of the virus saying, I'm not okay, I'm infected. Okay, the flag changes. Infection, cancer, I'm not okay. So antigen-presenting cells, B cells, macrophages, dendritic cells. Dendritic cells, most important antigen-presenting cells. So where can you find MHC again? Type 1 on all nucleated cells. Type 2 on antigen-presenting cells. Antigen-presenting cells, therefore, have both type 1, type 2. T lymphocytes produced in the bone marrow, mature in thymus, exposed to the antigen in lymphoid tissue, migrate in the circulation afterwards. Positive T cell selection. make sure that T cells can actually recognize, just see the flags. If they can see the flag at all, see MHC proteins, they can recognize MHC. So if positive selection doesn't work, T cells become useless. They anergic. They can't do anything. Negative T cell selection is to get rid of self-reactive T cells that attack our own tissues. So if this process fails, we have autoimmune responses. Two main functional types, CD4, CD8. Functional differences, CD4 differentiate into helper and regulatory cells. CD8 cells differentiate into cytotoxic lymphocytes. CD4 cells always recognize the antigen bound to type 2 MHC, and CD8 type 1. So that is demonstrated here. That's MHC restriction. Okay, right over here. Differences in, yeah, we covered this. So helper cells. That is a really good picture here. This one. So slide number 44. So basically, helper cells, they, first of all, they form a pool of memory cells, memory helpers. They also stimulate macrophage activation. They stimulate B cell activation. And they assist activation of cytotoxic lymphocytes. So now, you probably have finished or about to finish watching the videos for that unit, but you know that HIV infects CD4 cells. And as the result of this infection, CD4 cells, these ones, CD4 cells, they die. If you don't have CD4 cells, you have very limited B cell activation, you have very limited macrophage activation, and you have very limited CD8 into cytotoxic lymphocyte activation. So basically, all branches of your immune response are suddenly affected. Make sense? Like the entire adaptive immunity is wiped out. So how T cells link cellular and humoral immunity? Well, T helper cells activate B cells. How they say adaptive and innate? Well, T helper cells are necessary to activate macrophages, which is the branch of adaptive response. T regulatory cells, they are important to control immune responses, to dampen them so we don't have autoimmune condition. And in autoimmune conditions, we often see less T regulatory cells. So how do cytotoxic lymphocytes kill infected cells? By apoptosis. They can't phagocytose. So what's that system? Fast ligand, fast receptor. All I want you to know about this one is not where is what. The point is, you have cytotoxic lymphocyte, okay? Okay, let me go back to this. I hope you see me in the corner. You have cytotoxic lymphocyte, and you have a target cell. The receptor and the ligand interact, and that interaction initiates apoptosis in the target cell. That's it. It's like a killing machine, okay? Or they can release granzymes and perforins. The perforins will make holes in the target cell, and granzymes will enter the target cell through these holes and induce apoptosis. 5 types of the vaccines. We've got, that's all table. Now, in regards to this table, the vaccine table, what you need to know. So, most important, I want you to know the concept behind each of these 5 types, okay? Like, what is attenuated vaccine, or what is the DNA vaccine? Examples, absolutely no. Advantages and disadvantages, general idea. I'm not going to be too picky since, you know, it's very, 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 very specific. By the way, I strongly recommend you to open that link. Did anyone open that link? Well, I'm going to open it for you. That's how vaccines cause autism. That's how they do. Yeah, that's an actual website. Okay, that's what they cause. It's the main side effect of vaccines. So we got this one. Okay, type 1 let's talk about hypersensitivities really quick. I know, it's awesome. Absolutely awesome, yeah. I love this. I always say that the main side effects of vaccines is like being wrinkled and bald and having arthritis, That's a side effect because you're like 80 years old, something. Type 1 hypersensitivity, allergy. So what you've got there, you're exposed to the allergen first time, your B cells start to produce immunoglobulin E, which binds to the basophils, and that's it. Basophils, mast cells, that's it. You're exposed to the allergen second time, allergen binds to Ig that were produced before that, causes degranulation of mast cells, allergic response. Okay, symptoms, everybody knows what allergy looks like, so I'm not going to go into that. Vasodilation, runny nose, hives, blah, 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 blah. Anaphylactic shock. That's when basically like all mast cells degranulate at the same time, when there's massive vasodilation all over the body, which leads to precipitous drop in the blood pressure and constriction of the bronchioles, and you suffocate and you have cardiac insufficiency, vascular insufficiency, and you basically die. Type 2 hypersensitivities. Ugh. I mean, it's kind of fun, but ABO blood types. Ugh. Okay. Type O. No antigens, A and B antibodies. Type AB. Both antigens, no antibodies in the plasma. A, antigen A, anti-B antibody. B, antigen B, anti-A antibody. So agglutinins, those are antibodies. Agglutinogens, those are antigens. So if agglutinogens and agglutinins match, you have agglutination. Universal donor is type O because red blood cells do not have antigens. Universal recipient is AB because they don't have antibodies. Main symptoms of transfusion reaction is kidney toxicity, fever, occlusion of the blood vessels, so on and so forth. Rhesus factor. There are no naturally present antibodies against rhesus. So for instance, I'm rhesus positive, I don't have antibodies. If somebody is rhesus negative, you don't have antibodies either. So if mom is rhesus negative, child is rhesus positive, first pregnancy they find, mom delivers the baby, baby's red blood cells, when placenta detaches, baby's red blood cells enter mom's circulation, stimulate the production of antibodies. Okay, so now mom has antibodies against rhesus factor. Second pregnancy, mom is rhesus negative, baby's rhesus positive, but this time mom has antibodies. Antibodies cross placenta, get into the baby's blood, bind to the baby's red blood cells. Problem. So in order to avoid this, mothers, rhesus negative mothers, all of them, they receive ROGAM shot, basically external antibodies against rhesus factor, and those external antibodies, they bind to baby's red blood cells during the delivery and hide those red blood cells from mother's immune system. Okay, condition that results from that, if you don't do ROGAM shot, it's hemolytic disease of newborns. Type 3 hypersensitivity, IgG. Antibodies bind to the self-antigens, forming immune complexes, Immune complexes are lodged in the basement membrane. Macrophages, neutrophils attack immune complexes, destroy them, destroy basement membrane. You have inflammation in the endothelium, and that's bad. Like if it's kidney, it's glomerulonephritis. If it's blood vessels, it's vasculitis. Examples of type 3 hypersensitivity, lupus, and rheumatoid arthritis. Type 4 hypersensitivity. You are exposed to the antigen, well, allergen, let's say, allergen, and you have T cell response against it. So you form a bunch of T helper and cytotoxic lymphocytes. The most importantly, you form memory cells. The first exposure may come unnoticed. And then, when you encounter it second time, all those memory cells quickly proliferate and differentiate into effector cells. And now you have cytotoxic lymphocytes attacking the body site where the allergen is. And macrophages stimulated by T helper cells that attack that site as well. So that would be an example of poison ivy rash, really bad rash. It's called delayed because it's mediated by the cells, so it takes a while for it to just, manifest itself. Major types of transplants, auto, iso, allo, xeno. Remember one thing. Isograft is a transplantation between only identical twins. Fraternal twins doesn't count. Mechanism of rejection, the immune system of the host attacks the transplant. In graft versus questions? In graft versus host disease, the transplanted bone marrow from the donor produces white blood cells that attack the tissues of the host. So that's graft versus host disease. Immunodeficiencies, be able to distinguish congenital versus acquired. DiGeorge, no functional T cells, agamma-globulinemia, no functional B cells, SCID, no T, no B cells. Basically, you're screwed. Okay, let's talk about pathogens. Okay, I'm going to take a pause. I'm going to randomly ask questions. Darla?
No.
Good, okay. Nicole, any questions? We good?
Yep, we're good.
Okay,
Yep,
awesome.
we're good.
Okay, African sleeping sickness, microorganism, Trypanosoma brucei, transmitted by the tsetse fly, so it's biological vector. Signs and symptoms, fever, headache, myalgia, primary infection, then multiple organ failure, lassitude, neurological problems, and death. Geographical region, well, Africa. Disease is treatable. Vaccines not available. Chagas disease, Trypanosoma cruzi, transmitted by the sandfly, biological vector, signs and not sandfly. I mixed it up with leishmania. It's transmitted by the reduvid bug, the kissing bug. Yeah, the triatomins. Signs and symptoms, again, initially fever, then it can cause very specific symptoms. It can infect the heart and the smooth muscle, so megacolon, megaesophagus, cardiomyopathies. Geographical region, South America, disease treatable, no vaccine. HIV, RNA virus enveloped CD4 cells as the receptors. This is why virus infects mostly CD4 cells. Delta 32 mutation in the CCR5 gene in the host makes the host resistant to the HIV infection. Mechanism of the viral entry. So fusion, mostly fusion, so virus attaches to the receptor, viral envelope fuses with the cellular membrane, capsid gets disrupted. If viral RNA is released too early in the cytoplasm, abortive infection, it stops. Cell basically crushes it. Normally, RNA, after the release, gets reversely transcribed by the reverse transcriptase into the double-stranded DNA. And the double-stranded proviral DNA is incorporated into CD4 cell chromosome, CD4 cell DNA, by the enzyme called integrase. So that's the so HIV that's incorporated into cellular DNA is called a provirus, and provirus may not be expressed, and that is a latent infection, but eventually it gets expressed, produces viral proteins, copies of viral RNA. RNA is packaged into the virion, but the virion requires proteolytic processing, so protease activity is required for the HIV virion maturation. During the course of HIV infection, CD4 cell counts drop, patient has no immunity, and a bunch of secondary infections lead to the patient's death. So continent that has the highest HIV prevalence: Africa, the lowest, surprisingly, Arabic Peninsula, Main transmission routes. When I say main, I don't know if you've listened to the lecture. I give some pretty wild examples. Like HIV, there were recorded cases of transmission during kissing, but it was violent kissing that involved, I guess, a lot of biting, But the main transmission routes are sexual and use of the sharing needles. Okay? No blood transfusion. Vertical, not in the United States, but vertical transmission in Africa is really big, from mother to child. Best strategies to prevent the transmission: needles, distribution of needles, safe sex, and treating people with antivirals. Socioeconomic groups: poor. Prevalence in the United States, the disease affects mostly African American population, also a Latino population. But if you would look, I have that lovely picture. There's a graph showing here that once you take poverty, like just look at the people with the same income. Basically, if you're poor, there's a huge percent of HIV positive. If you look overall, it's mainly African Americans, because African Americans, more of them live in poverty. Okay? So, steps of the life cycle that are targeted by modern antiretroviral therapies. So you've got attachment and fusion. You can inhibit reverse transcription, you can inhibit integration, and you can inhibit proteolytic processing of the virion. So life expectancy for the HIV positive patient on the heart is basically the same as for patient without HIV. And resistance, that's a real important question. How do you prevent resistance to antivirals? HIV mutates very quickly. So in order to avoid resistance, we use antivirals in combination. Because for the virus, it's fairly easy to evolve and adapt to a selective pressure from one drug, but if you use 2 or 3 simultaneously, it's almost impossible to adapt. So we change the regimen courses and we use combinations. That's how resistance is prevented. Major challenge for the development of HIV vaccine: it changes very fast. Okay, so it's a big problem. And also, if you think about it seriously, like if you get a vaccine against, I don't know, mumps, and it's not 100% effective, and you still get mumps, I mean, you're going to be upset, but you're not going to die. If you get a vaccine against HIV and it's not 100% effective, and you get infected, that's it. You have HIV for life. Like, ideal HIV vaccine should protect everybody, ideally. Otherwise it's kind of, So that's the challenge. Okay, I'm going to stop the recording.